suppose. So kindly allow me to carry on there. So Mr. Siddharth, now
Yes,
you
sir.
can proceed. I suppose if guests are allowing me. Professor Sharma sir, may I allow? Continue.
Namaskar and most welcome in 4th annual meeting of International Association of Zoologists IAJ and International Virtual Seminar on Recent Trend in Life Science and Biotechnology are organized by International Association of Zoologists and Siddhi Artha Group of Education are collaborate with Department of Biotechnology, Maharishi Markandeya deemed to be University Mullana Ambala, Sharda Institute of Science Technology Education and Research, and Organization for Conservation for Environment and Nature. I express my heartfelt gratitude to the Honorable Sri Tasreem Kumar Garg, Chancellor, Maharishi Markandeya deemed to be University Mullana Ambala, Professor Ashok Kumar, Principal Engineering College, Mullana. I am also thankful to respected co-chairman and the backbone of the conference, Professor Anil Kumar Sharma, formerly postdoc and senior research scientist, UIC, Chicago, USA, and head of the Department of Biotechnology, MMDU, Mullana. In the other word of Shakespeare, I can no other answer make than thanks and thanks. I would like to thank the most important person of this conference, Dr. Sushil Kumar Upadhyay, President, Sister, and Honorable General Secretary, IAJ, and Assistant Professor, Biotechnology, MMDU, Mullana, Professor Raj Singh, Department of Biotechnology, MMEC, deemed to be University, Mullana, as a convener. I am also thankful to an organizing secretary, Ms. Mahasweta Mandal and Ms. Mansi Girdhar. Last but not least, I am grateful to Department of Biotechnology, Government of India, Council of Scientific and Industrial Research, Government of India, Defense Research and Development Organization, Ministry of Defense, Government of India, All India Institute of Medical Science, Jawaharlal Nehru University, Delhi, for their invaluable aid in knowledge matter related to the event. Medical profession, be it doctor, nurse, or pathologist, as well as biotechnologist, would not be possible without the study of biology. Pathologists identify disease by studying tissue, matter, and cells. Pharmacologists are responsible for creating and developing different drugs and help us medicine to combat disease, while nurse and doctors are there to ensure proper diagnosis and help us recover from various disease, illness, and various sickness. But biotechnologists are the combination of both three. This field are especially backbone of the advanced medical science and research diagnosis. On behalf of the organizing committee, I am grateful to all the guest speakers and number of experts visiting from different educational institutes for sharing their experience, for making the seminar popular to attract scholars from all over the world. I extend my profound sincere gratitude to our esteemed chief guest and keynote speaker, Professor Sandeep Kumar Malhotra, formerly head of the Department of Zoology, University of Allahabad, Prayagraj. From this consent and support, I am grateful to our guest and speaker, national and international, to accept our invitation. We are very grand welcome to our chief guest and keynote speaker, Professor Sandeep Kumar Malhotra, D.Sc, ACU, Prayagraj, Professor Anil Kumar Sharma, Head of the Department, MMDU, Mullana, Dr. Himangsu Ojha, Scientist E, Department of CVRN, INMAS, DRDO, Delhi, and Professor B.S. Balaji, School of Biotechnology, JNU, Delhi. Without taking more time, I would like to invite Professor and Head of the Department of Biotechnology, Dr. Anil Kumar Sharma, for his blessing and kind word about conference. Thank you.
Most welcome.
A very good morning to each and everyone. And I am extremely proud to know that this fourth meeting of International Association of Zoologists and an international virtual conference seminar on recent trends in life sciences and biotechnology is being organized by the International Association of Zoologists and Siddhi Artha School of Education in collaboration with our Department of Biotechnology, Marci Markandeshwar deemed to be University Mullana. Moreover, what I find is the theme of the conference, that is on biomolecular immunology in wound healing and cancer development, strategies to combat human diseases. It's also quite apt, keeping in view of the current scenario as far as diagnosis in cancer is concerned. Sincere congratulations to the International Association of Zoologists and the other organizers, including Siddhi Artha Group of Education, Sharda Institute of Education, Science, Education and Research, along with organizations for conservation of environment and nature, for organizing this event. And I profoundly wish for the success of the program. At the outset, I would be highly, extremely thankful to the esteemed management of all the collaborating institutions and the other organizers also, along with all the esteemed dignitaries of the various campuses involved in this organization of the program. Let me profoundly welcome and thank Professor Sandeep Malhotra, who is the Professor of Eminence, a person par excellence, who is currently at the ACU Prayagraj, who is also the chief guest and keynote speaker of today's function. I also wholeheartedly welcome and thank all the distinguished speakers, including international and national my congratulations to the convener of this event and the whole organizing team for bringing this forum of intellectuals together at a single platform through this virtual seminar. And I profoundly wish for the success of this international seminar. Once again, I profoundly welcome you all with immense joy and pride, and wish for the grand success of this particular seminar. Thank you again, and back to Siddharth for the next proceedings. Thank you.
Thank you. We are very great welcome to our chief guest and keynote speaker, Professor Sandeep Kumar Malhotra, D.Sc, ACU, Prayagraj. And thank you, Head of the Department of Professor Anil Kumar Sharma. Thank you so much, sir.
Just a minute. I'm enough lucky to have here my Reverend Guru, teacher, Malhotra sir. And without his blessing, it was not possible to be here in such a way to organize this kind of things. He has guided me at each and every corners of academia, even though livelihood.
मम्मा शोर ना करो आप लोग, मैं सुन रही हूँ, मेरी सेमिनार चल रही है.
Our department, who always encourage us here, Professor Adesh Saini sir, and now it's a matter of indeed proud and privilege to me here with Professor Malhotra sir, my guide. So please sir, Professor Malhotra sir. Sir Malhotra sir, over to you sir.
Okay, great. Dr. Sushil, good morning. आप जरा चेक कीजिए, स्लाइड शेयर हो रहा है?
Yes, sir.
Please share the slides. Slide share कीजिए.
Allow sir to share the slide.
हाँ मैंने किया है. स्क्रीन शेयरिंग कीजिए. You have disabled. आप इसे एक्टिव कीजिए. Kindly allow my screen sharing.
Yes, sir. Yes, sir. Yes, sir. Just a second, sir. स्क्रीन शेयर के लिए सिद्धार्थ.
Yes, sir. Multiple participant can share simultaneously is on.
I have shared my screen.
या
Yes,
सर
sir.
को आप अलाउ करो. आप किसी को कॉल रिसीव ना करो. आप सर का स्लाइड शेयर करना स्क्रीन करो.
Yes, sir.
अलाउ करो सर को. जी.
इधर ही तो आगे कभी पीछे जा रहा है.
Try once again, sir.
क्लिक करना क्लिक. अच्छा दिख रहा है ना. ठीक है.
कर दिया.
Yes, sir.
नहीं म्यूट किया.
सर
नहीं आया नहीं. Mine is already on.
म्यूट किया है.
okay sir. Once again, sir. Try sir once again. Go through share screen and then.
हाँ एक मिनट होल्ड कीजिएगा. होल्ड कीजिए. It has come. Is it visible?
Yes, sir. Now is. Yes, sir.
Okay,
Visible,
okay.
sir.
Yes,
Okay,
sir.
okay. So good morning, Professor Sharma. Thank you very much for the nice piece of introduction. And I know the hard taskmaster Dr. Sushil is. You have been put up very nicely. And it's a grand thing to see the institution growing, that you're able kind of activity.
Visible, sir.
आप इसे अनम्यूट कर दीजिए सबका. सबका माइक अनम्यूट कीजिए, नहीं तो यहाँ आवाजें आती रहेंगी.
जी सर. Muted.
Okay, sir. Okay. Thank you, sir.
जी म्यूट कर दीजिए. सॉरी. माइक.
सर आपने खुद को म्यूट कर लिया. इसको अनम्यूट कीजिए प्लीज.
Sir, please unmute yourself, sir. Yes, sir. Yes, sir.
Okay.
Yes, sir. Right now, okay, sir.
जी. तो ये बात मैं कह रहा था कि कार्सिनोमा कई तरीके का होता है और मोस्ट कॉमन काइंड ऑफ थिंग्स यू नो ऑलरेडी. Because of limited time, I would not be able to proceed with those things which are commonly known in the medical science. We are more concerned by the helminth, the parasitic helminths, which are the ones reside inside our body organs. And in addition to parasitism, they directly damage the development, health, and prosperity. Now, where do they actually reside? Practically every part of the body, हर जगह ये मिलते हैं शरीर के अंदर और इनका मोड ऑफ ट्रांसमिशन इंटरमीडिएट होस्ट के थ्रू आप जानते हैं. Our point in study is experimental studies in animals have confirmed the carcinogenic potential of these parasites. Pathogenesis though remains unclear. अभी ये nascent stage में है science, बहुत ज़्यादा इन चीज़ों के बारे में मालूम नहीं है, लेकिन the initial carcinogenic event is probably a function of the length and severity of infection. जितना समय तक होगा और जितनी intensity इसकी ज़्यादा होगी, उसका प्रभाव पड़ने की ज़्यादा संभावना है. So the host immune response and such variables as ingestion of dietary carcinogens, this is the primary causes. This is the organism Opisthorchis and Clonorchis. ये bile duct में रहते हैं, trematodes हैं. They travel through snail intermediate host in the aquatic bodies. और भी जानवर हैं जिनके शरीर में ये मिलते हैं. Human beings में इनकी मौजूदगी चिंताजनक है. They cause cancer of the bile duct, which is known as cholangiocarcinoma, and body parts trematode का classroom studies में आप करते हैं इनको. Opisthorchis and Clonorchis parasitize the bile ducts of millions of people. The most important aspect of infection with these flukes is their ability to initiate cancer. Otherwise, they would not have disturbed us. The numerous studies have shown these flukes to occur in association with cholangiocarcinoma far more frequently than can be explained by chance. बहुत ज़्यादा इनको देखा गया है हाल के दिनों में. Opisthorchiasis caused by Opisthorchis viverrini and Clonorchis are etiological factors in the causation of human bile duct cancer, especially cholangiocarcinoma. So once we are convinced कि ये world के किस part में बहुत ज़्यादा है, it is Thailand, northeast Thailand का area जहाँ अभी मैप मैं आपको देता हूँ जिनमें ये देखने की जरूरत है कि चाइना, कोरिया, जापान होता क्यों है ये. Actually these parasites require 2 intermediate hosts और जहां से ये सर्केरिया निकलकर बाहर आते हैं और फिर ये मेटा सर्केरिया में कन्वर्ट होकर मैमेलियन होस्ट को ढूंढते हैं. तो जब आप अंडर कुक्ड या अनकुक्ड मीट को लेते हैं, फिश को खाते हैं, उनके माध्यम से ये आपके शरीर में एंटर करता है. तो मेटा सर्केरिया एक्सिस्ट in the duodenum and the juvenile flukes migrate using their 2 suckers as locomotory organs through the ampulla of Vater into the common bile duct and extrahepatic biliary system. बहुत unusual है इनका hepatic system तक पहुंचना और bile ducts में मौजूद होना. यही वो चीज है जहां पर migration की अहमियत समझ में आती है. Only because their developmental stages can have the potential to migrate, they reach the bile duct where they reside and cause cancer, where they mature and commence egg laying after 3 to 4 weeks. यहाँ पर इनकी संख्या जब बढ़ती है तब ये disease cause करते हैं. Clonorchis and Opisthorchis infections may persist for many decades, and light to moderate infections, a few hundred worms, rarely produce significant clinical symptoms. बहुत ज़्यादा ये detect भी नहीं होते. अगर इनकी मात्रा कम है तो कोई disease ऐसी cause भी नहीं करते. इसलिए ये बहुत harassing है कि जब इनका संख्या मात्रा में बहुत ज़्यादा बढ़ जाए, उस समय ये हमें बीमारी का पता चलता है. Unlike with hematoma, there appears to be no link between cirrhosis and primary cholangiocellular carcinoma. There are major differences between the age and sex of patients dying from hepatocellular carcinoma and from cholangiocarcinoma, suggesting that different etiological factors are operating for each. Now, branches like these are not flourishing in many parts of the countries. They are aware only, helminthology is the science which is very great, having great potential, but people don't care much about these things because the diseases most of the time cause debility and they are not so harmful. They do not produce such a grave danger as you understand the cancer is. Therefore, they are neglected. So presence of liver fluke infection in a population with high incidence of primary liver cancer is associated with overall increase in the incidence of carcinoma. And this is the part of the world where they are found the most. Southeast Asia, showing the area not very far from our own country. So these are the things where the habits actually, when we take uncooked fish, we are very fond of consuming raw fish. And most of the time even when we cook fish, part of these remain uncooked. And when you do not leave bile ducts or gallbladder or these part of things from your cooking table, then there is a greater danger for spreading the disease. So larger second order bile duct in the hamster containing an adult Opistharchus viverrini, showing the bile duct epithelial hyperplasia. You can see hyperplasia there, and polypoid formation, dense periductular inflammatory infiltrates. This is inflammation, which is the graver cause of this disease or the after effect of the disease. Then the proliferation of bile duct tubes with lumen formation in a hamster infected with 50 metacercariae. You understand, once it establishes, it tries to spread its tentacles all over the infected tissue. Proliferation of spindle-shaped bile duct epithelial cells. You can see the arrow is marked for showing the cord-like structure, which is where the proliferation is going on, epithelial cells, with piecemeal necrosis of vacuolated hepatocytes in an ovivirini infected hamster feeding a low protein diet. Note the sharp line of demarcation between proliferating biliary epithelial cells and hepatocytes. So these are the structural changes that the pathogen causes. Chronic infections caused by certain pathogens such as virus, bacteria, fungi, and parasites contribute to about 18% of the global burden of cancer. Helminth infections attributing to only a small part of it. Carcinogenesis associated with helminthic infections induced development of cancer is a very complicated event involving several different mechanisms, varying from one species of parasites to the other. ROS involvement and other kind of things, RNS, you are very familiar with these things, but since their infection is very lesser, the details of these have been worked out in smaller number of studies. Parasite infections evoke immune responses in the host, which finally result into inflammatory reactions. I told you the inflammation is the primary cause that gives rise to cancer or produces a potential danger. The chronic inflammatory processes produce ROS and RNS. These active species, these free radicals may cause DNA damage, resulting into genetic instabilities and occurrence of malignancies. We did try on a separate note while we work with a single cell biology. This is our photograph of DNA during mitotic division in the liver cell suspension of a fish parasite. So you can clearly see the division of the DNA, and that is damaging. It is not helping the human beings to fight out with this kind of disease. And once DNA is damaged, the kind of genetic activities are disturbed. Third one is schistosoma hematobium, and its invasion occurs into blood vessels. You know, this is a hematoparasitic element. This organism invades through the tissues and causes local inflammation, regeneration process, and cell proliferation. This is the mechanism how schistosoma hematobium infects. DNA alterations are done here, and metaplasia or hyperkeratosis are the ones, the ultimate results. Third or the fourth infection by spirocerca. So now you will have some idea. These are very small, unrecognized organisms which are called parasites, but there is a huge number, great variety, even Taenia solium. So I'm showing you the mechanism of spirocerca. These are the nematodes, and you can clearly see they are residing in them. You understand this reside there for millions of years and thousands of years until humans are dying, and then it is being passed to other organisms, then third generation. So it continues. So the nematodes are very difficult to get away with, and ultimately they form nodulations in this kind of developmental mechanism. Esophageal cancer they cause, spirocerca, which is a nematode. Tissue section, fibrosarcoma. So fibrosis is a common one as it happens in man. In man, the histopathological response to infection with Opistharchus or Cleararchus is distinctive. They're very distinctly infecting, and you can identify those infections separately. The infected liver revealed slight irregularities at the capsular surface in the mild to moderate cases. But in severe cases, pale cystic areas can be seen where the fibrosed bile ducts approach the surface. Main issue is fibrosis, and in this case, it is occurring in bile duct. The cuts revealed large and medium-sized bile ducts, 2 to 3 times their normal size, with walls several times thicker than normal. All tissue reactions in a similar way as it happens in mammary glands or other tissues of human beings. The early stages of episarchiasis and clonarchiasis are both characterized by bile duct epithelial proliferation, followed later by periductal fibrosis. Biliary hyperplasia is the distinctive lesion of early liver fluke infections. Infections by Opisthorchis and Clonarchus and Schistosome are classified as group 1 biological carcinogens. You call them biological carcinogens, definite causes of cancer. And these worms are metazoan eukaryotes, unlike the other group 1 carcinogens, including human papilloma virus, hepatitis C virus, and Helicobacter pylori. Other things why they give us an insight why is infection with only a few helminth species in the form of carcinoma. Not every parasite causes cancer, and this is the question that leaves us perturbed. Why only a few ones have the potential to cause cancer? Let us examine carcinogenicity of these helminthic infections, including roles for catechol, estrogen, and oxysterol metabolites of parasitic origin as initiators of carcinogenesis. The liver fluke Opisthorchis is endemic in Thailand, Laos, PDR, Vietnam, and Cambodia, whereas Clonarchus is endemic in Vietnam, China, South Korea, North Korea, and far eastern Russia. Not very far off is the country where we reside, and things are to be examined in detail before we get hold of our own area in part of the country the infection is most prevalent. Opisthorchiasis results from ingestion of metacercarial stage of Opisthorchis. Insisted is in undercooked freshwater cyprinoid fish that we consume very frequently. Related Opisthorchid flukes, sinensis, felinius, which like viverini, also are transmitted through consumption of, so the source of infection is the same consumption of undercooked fish. Following ingestion, the immature worms migrate from the duodenum to bile duct, where they mature into adult forms within the lumen of bile duct and gallbladder. So they migrate within the body, and parasite eggs are shed into the bile, pass out to the environment in the fecal stream. Freshwater snails ingest the eggs. How the cycle is being completed. The parasite eggs hatch in the digestive tract of the snail, and larval miracidium migrates to hepatopancreas of the snail. That is within the intermediate host. Asexual reproduction through sporocyst and rediae stage follows, culminating in release from a snail of a motile aquatic larva termed as cercariae. That is the dangerous form, and cercariae are the ones that affect human beings, so the final host, whichever they are. These in turn release aquatic cercariae that penetrate the flesh of fishes that are the second intermediate host of fluke's developmental cycle, and continuous deposition of eggs of S. hematobium in the bladder can lead to squamous cell carcinoma. That is about your schistosoma. That is a picture of how life cycle is completed, which I just told about. And things are readily available in classroom classes, textbooks. They are not very difficult to get hold of. Only thing new is that they have the potential to cause carcinoma. The melt profiles, now that was done on RT-PCR by us. How do we differentiate the species if they are such minute organisms that we cannot differentiate even species of Dacnitoides, Anisakis, Contrasecum? They are of zoonotic significance. Anisakis is the most dangerous one, occurring in Indian fishes. And I deviate a bit from direct DNA damage to the DNA division into single cell, single component of DNA. That is how what we understand the melt profiles or the melt peak curves when the DNA is getting affected. Ultimately, by the RT-PCR process, we examine the melt peak curve when actually the DNA fiber is separated. the twin elements which hold together, they get separated. That point of melt peak is different for every parasite. I make it to be understood clearly that DNA helix is being disentangled at higher temperature, and that temperature varies from species to species. So once we understand what is this, then we understand the differentiation of species to the molecular level, and that is what is achieved by RT-PCR. This was published by us in Single Cell Biology. You can understand the peak. Figure 1 shows single, figure 2 shows variation in the single, figure 4 shows double peak. So you can understand parasite-wise things are varying, and they are all parasites of Rita. That simultaneously, molecular analysis by sequencing. You understand ITS, ribosomal DNA, all the things which we examine through nucleotide analysis go simultaneously. And this study only helps us to understand it more precisely, why molecules are different in Asexual from Contrasecum, from Decnitoides, and that is how the diagnosis is continued to understand differentiation at the species level. You will see all the curves different, and this is the profile of multi-peak curves. The aligned melt peaks, you can understand single, even triple layers, and all larval and adults are showing similar form of, no significant variations, but genus and species-wide variations are very much there. So this is what we try to conclude, that molecular diagnostic procedures based on RT-PCR have proved to be reliable biotechnological tool from the standpoint of segregation of species and other taxa. For years together, we continued segregation study on the basis of morphology, and people put a lot of questions. Why do these are not reliable parameters? So we got down to nucleotide analysis, and then we further got down to DNA helix separation and single strand analysis. On the basis of melt peak profiles, we can differentiate between the parasitic forms, which are very, very minute, and from the naked eye, from microscopic eye, you cannot differentiate. Their potential to detect multiple pathogens simultaneously in one setting has put these on a higher pedestal of clinical significance as well. The asymptomatic infections of invasive parasitic protozoans were detected in earlier investigations. The inquisitiveness about such infections could thus be PCR-coupled technology, has been scarcely used for detection and diagnosis of helminth infections, particularly of zoonotic significance. You understand zoonotic significance when a parasite travels through an invertebrate intermediate host to a human being, that is called a parasite of zoonotic significance. हर पैरासाइट ह्यूमन बींग्स को अफेक्ट नहीं करता, ये बहुत समझने की चीज है। केवल कुछ ही ऐसे हैं जो जूनोटिक सिग्निफिकेंस के कहलाते हैं, और इनसे पैरासाइट से ह्यूमन को खतरा है। ऐसे भी हैं जो समुद्र के पानी में मिलती मछलियों में पाए जाते हैं और वहां से ट्रैवल करके गंगा तक आ गए हैं। तो इन चीजों के बारे में मॉलिक्यूलर लेवल सिमिलैरिटीज ही ये स्थापित कर सकती हैं कि हमें इनके बारे में सतर्क रहना होगा। इट इज नॉट ओनली एन इन्वेजन ऑफ द होस्ट fish, but it carries parasites from the seashore areas to the fresh water, and that is what is dangerous. Therefore, the diversity of sequence variations within and among samples can be determined by melting point analysis. Hence, the distinct identity of individual amplicons comprising contrasting sequences could be established by the use of melting curve analysis as a tool.
Thank you, sir. Thank you.
Threshold cycles. Asimplex, that is Asexual, and Decnitoides assays were consistent over time. So these are the brief temperature-like substantiation for different species. You can find these strand separation at these temperatures. I think that is where do I conclude. And thank you very much for having given us the platform.
For this kind of elaborative task, thank you very much, sir. We are very much enlightened.
Hello.
And I request Malhotra sir, Professor Professor Sharma sir. I request Professor Sharma sir to felicitate Professor Malhotra sir by the fellowship. सर प्लीज आप शेयर स्क्रीन को हटा दें सर थोड़ा सा तो on behalf of IAZ and organizing committee, I wish to request Sharma sir, please felicitate Professor Malhotra sir by the fellowship of International Association of Zoologists.
So many congratulations, Professor Malhotra, for such an elegant talk and for this honor for being the Fellow Award Certificate from the International Association of Zoologists. I congratulate wholeheartedly for this wonderful achievement, and many more laurels also you have achieved in your career. This one is another one added to the cap. So many congratulations, Professor Malhotra.
I'm happy.
Thank you so
I'm
much.
happy, Dr. Sharma. Professor Sharma, thank you so much. I am indeed obliged. My best wish is that the flowers are blossoming from Allahabad to your place, Mullana, and
you,
under
sir.
your guidance and stewardship, they will be carried far ahead. Thank you so much for the honor.
Thank you so much. Thank you, sir. Thank you.
Thank you so much, sir. और हम लोग नेक्स्ट सेशन प्रोफेसर शर्मा सर का सुनेंगे. जस्ट आफ्टर 5 minutes we will start the next session on the same link. So our next speaker will be Professor Anil Kumar Sharma sir, Professor and Head Department of Biotechnology.
Excuse me, sir.
Yes,
I'm Dr. Neelam Mattu. Yeah, can I talk to Dr. Sandeep Malhotra, please?
जी मैम.
Yeah, kindly connect.
Okay, मैम.
Hello.
जी मैम.
Namaskar, Dr. Malhotra. I don't know he is listening me or not.
I'm here, Neelam.
Okay, sir.
Yeah,
namaskar, sir. Very warm greetings to you. Though we are connected on social media, but meeting you for the first time virtually here. So congratulations for this award and also for the brainstorming talk. Actually, such a talk is the need of the day. And I know that how much hard work you have done throughout your life. And you very well deserve such awards. And it was really great hearing to such a distinguished scientist. I would just like to say 2 words for your hard work. Uddhav Menahi Karyani Naman. Yes, sir. Uddhav Menahi Karyani Naman Nahi Chuptasya Se Hasya Pravishati Mukhe Mriga. That is, hard work is the only key to success, which